Hello everyone. Today what we're going to talk about in this podcast are some alternatives to Mendel's predictions. And we're also going to talk about pedigrees. I want to begin by saying that when Mendel first made his discoveries in the 1860s, it was quite revolutionary what he was able to put together. But the interesting thing was nobody knew he did this until 1900. His works were published, but they were published in an obscure journal, and it's hard to know how many people read it, but probably very few. And it wasn't until 1900 that other scientists, independently discovered the same numbers. And to their credit, they gave Mendel credit because they discovered his work in this obscure journal. But they were nice enough to give him credit for first discovering it. There's actually a deeper story to it. One of the scientists actually didn't want to give him credit, but that's for another day. So, shortly after his works were rediscovered, and he had already passed away by the time his works were rediscovered, but right after they were rediscovered in the 1900s, a lot of geneticists, a lot of scientists, most of them at the time were botanists, but not all of them, were trying to see if Mendel's rules applied to their own special plant or organism that they were working for. And by and large, what they discovered was that Mendel's works, his predictions, held true with all the traits they were looking at. Now, every now and then there came an exception. And it turns out the exception was his rules still were true because his rules really are based upon the inheritance of the chromosomes. But sometimes the manifestation of those genotypes appeared differently in the phenotype. And that's what became different. So these are commonly referred to as the exceptions to Mendel's rules. The first one your book talks about is incomplete dominance. So what is complete dominance? Well, let's first begin by asking what complete dominance is. So for instance, with complete dominance, you might predict you'd have a red flower. Let me color this in red as much as I can. And if you crossed it with a white flower, again, if this were complete dominance, what would happen is if you cross these together, what you would get is what? If it was complete dominance, what would it be? Well, if red was the dominant allele, then this would be red. If white was the dominant allele, it would be white. Typically, in these kinds of crosses, if it's complete dominance, you would get a red. Usually the darker color is the dominant color. So you get a red flower. Now, sometimes what would happen, when you cross these together, instead of getting one or the original phenotypes, you would get pink. A pink flower. And this was really interesting to them. How did you get pink? And what was even more interesting is if you took another pink flower from the same cross here, the progeny from it, and you cross-fertilize them, what do you get? So if you cross these together, what are the results? You might predict you'd get all pink. That might be one prediction. You might also predict red would come back and it would be all red. Or maybe you'd predict they'd all be white. Well, what happens is when you cross these together, 50% of them are pink. 25% of them are red, and 25% of them are white. And I'm not going to show it here, but if you take this red one and you cross it with another red one, they're all going to be red. So it turns out this is a true breeding red flower. And this is a true breeding white flower. This other one over here, the pink one, when you cross it to itself, once again, you get a 50% pink, 25% red, 25% white. So how is this possible? This seems to counter some of Mendel's predictions. Well, it turns out it doesn't really counter it at all. It supports it because Mendel's prediction, let's go to, now this over here is a cell. It's not a plant over here, a flower. This is a cell. Let's think about what's happening in this cell. Let's say this cell has 2 chromosomes in it. I'm going to draw them this way here. 2 chromosomes. And let's say there's a gene in here. I'm going to move this across like that. And there's a gene in this one over here. And let's say this gene codes for the color of the flower. And let's say, and remember, let's remind ourselves of the term allele. These are different alleles. This particular allele, this version of the gene, is going to be responsible for red color. Okay. And if both of them are red, and you go through meiosis, this one's going to contribute to the red color, and so will this one. Let's go ahead and draw another plant over here. And this one's, we're going to have it being a pure breeding white flower. And so the gametes are, or the chromosomes here, they contain the same gene over here, but in this case, it is the white color allele for the flower. All right. Now, when this makes gametes, this one here, let's hope I don't run out of room here. It makes gametes. All of its gametes are going to contain the chromosome here. The gametes here, they're going to contain the red color allele. And this true breeding white color flower is going to produce gametes, and only gametes, with the allele that has the white color. Now, if these cross-fertilized here, you'd get all plants with red, and if these two cross-fertilize, you get all plants with white flower. But if they mix them together, what you're going to get is this gamete is going to fuse with this gamete. All right, so if you mix them together, you're going to end up with progeny that has the 2 chromosomes. One comes from this parent here, and one comes from this parent here. So you end up with a heterozygote, where it contains the red allele on this chromosome and the white allele on this chromosome. We call this incomplete dominance, the phenotype we see here, the pink here, because neither one of these is fully dominant. They're incompletely dominant. This still produces half the red, and this produces half the white. And when you mix a red and a white together, if they're both expressing it fully at these 2 different colors, you get pink. And so that's why this plant, when it becomes grown, it has pink flowers. So Mendel's rules still apply. His first rule says that these 2 gametes are produced by the separation of these 2 chromosomes. Each gamete gets one or the other. Same here. Then when the progeny, they're formed by a combination of these 2 gametes, and so the combination of these 2 chromosomes. Everything's fine. Now, what happens when we cross-fertilize? Well, let's first draw our gametes. In fact, what I'd recommend you doing, just for practice, is going to hit pause on the video here and make your own predictions. What's going to happen is, through meiosis, each one of these gametes are going to get one of these chromosomes. 50% are going to be red, and 50% will be white. When they fertilize, when you cross them together, you get, well, why don't we do this? Let's draw a Punnett Square, because this is fun. Let's draw a Punnett Square here, and we're going to put the white allele here, so let's just write W, and W here, and the red allele here, and the red allele here. If we fill out the Punnett Square, what you're going to find is we're going to see these numbers over here. So let's go ahead and do that. This red and this red come together. You get a homozygous red alleles, 2 of the red alleles, similar to what we see here. The plant will be red. You also get a red and a white. And when you have one of each of these, this is pink. So this plant here is a red and a white. This here is RW, and it's going to be pink. And then you have the WW. It's homozygous for the white allele, and so it's going to be white. Again, what we get are 25% of them, this big R, is this one here. 25% of them are WW, right here. And then 50%, these 2 here, are going to be pink. If I ask you for the phenotypic ratio in a problem like this of something that was incompletely dominant, you would say it's 1 red to 2 pink to 1 white. If you don't know, if you're given a problem and you're not told it's incomplete dominance, the easiest way to tell is by looking at the progeny of the first cross. When you take the two homozygotes here, homozygous red, homozygous white, you cross them together. If the progeny is a blending of the phenotypes, so pink would be a blending of red and white, then you know it's incomplete dominance. It's complete dominance if one or the other, red or white, would have been found in the progeny. But since it's a blend, we know it's incomplete dominance. Color is often used to explain incomplete dominance, but there's also conditions in humans that we see this. For instance, there's a cholesterol disease called hypercholesterolemia. And in this case, let's see, why don't we just write it here? We have space here, why not use it? In this case, if you're big H, you have healthy levels of cholesterol and you can metabolize it. If you're little h, you have the disease of hypercholesterolemia. And these individuals will have insanely high cholesterol even as a child, in the neighborhood of well over, say, 200 or 300 when you're a little child. These individuals almost always die at a very, very young age. The heterozygote, well, actually, let me take this back. If this was complete dominance, what we would say is, what we would predict is that the heterozygote here would have healthy cholesterol. But if it's incomplete dominance, it's going to have a phenotype that is somewhere in between healthy cholesterol and lethal cholesterol levels. And that's exactly what we see. We find people that are heterozygous to be somewhere in between. They're not healthy, but they're not going to die at a young age because of increasingly scary high levels of cholesterol. They typically will have 200 or so levels of cholesterol. They'll be on medication. It's very treatable. So it's a phenotype that falls in between these two extremes. All right, let's go ahead and move on to the next slide and talk about the next example of an exception to Mendel's predictions. In this example, it's called codominance. It's kind of easy to get codominance and incomplete dominance mixed up. So if you do, don't feel bad. But let's work on it so we can make sure that you understand it. Probably the best example to understand this is to look at blood groups. And let's just talk about A and B, because that's going to feed in nicely with another example of multiple alleles. There is a gene in humans, it's called the I gene, and it exists in multiple forms, either the A allele or the B allele. We're going to focus on these two first, but there's also another allele called the I allele. But what we're going to call it is the O allele. Sometimes you'll see it referred to as a little I, but most people are used to the terms O. All right, but let's just look at A and B. If you have, and I'm going to, I'll do it this way. If you have a person that is IA, and they're homozygous for IA, their blood type would be A. Now, if you had somebody else that was homozygous for blood type B, so phenotypically they are blood type B, like so. Now, if you took these two individuals and they had children, this person here would make gametes that only contain the I allele. And this person here would make gametes that only contain the B allele. So when they produce children, like so, circle them so you know that I'm talking about them together now, they exhibit what we call codominance. To understand that better, let's think about if this was a dominant phenotype, how we would look at the phenotype. If A or B was dominant, that is, if A was dominant over B, this person's blood type would be A. If B was dominant over A, they would have blood type B. But we know that neither one of them is dominant or the other. They're also not incompletely dominant. They don't share the phenotype like you see in incomplete dominance where you get this blending. They are both fully dominant, meaning this cell is fully dominant A. Well, let me not say it quite that way. This cell has the phenotype of A. It also has the phenotype of B. It's not blended, and neither one is superior over the other. And because of that, we don't say they're blood type A or blood type B. We say they're blood type AB. A and B are codominant. They share in their dominance. Now, if somebody who is AB had children with somebody else who is AB, they're not plants, so we can't self-fertilize, but if you had two people that were each AB, what you would do is you'd fill out your little Punnett square here. For simplicity, I'm just going to write A here and B. But for instance, remember that this person, when they create gametes, either sperm or eggs, half their gametes are going to be A type and half their gametes will be B type. If their partner also was AB, half their gametes would be A and half their gametes would be B. To figure out what the phenotype of the children will be, or the possible phenotypes of their children, we have an A and we have an A. We have a B. We have a B. Oops, I said that wrong. We have an A and a B. I got a little ahead of myself. We also have an A and a B here. If you're uncomfortable or don't understand how I'm doing these Punnett squares, hold on a moment and let me walk you through this. So remember, Punnett square is just a tool we use to make predictions about what two parents, what their progeny will, how they will appear. So on the top axis, we will put the gametes from either mom or dad. And then on the vertical axis here, you'll put the gametes of the opposite parent, A or B in this case. So this parent produces A gametes and B gametes, blood type that is, and same here, A and B. Then to determine what their phenotypes are, you unite this gamete A with this gamete A, and you end up with AA. You unite this gamete A with this gamete B, AB. This gamete B with this gamete A, AB. This gamete B with this gamete B, BB. All right. We could do this with any trait, just like you saw in the previous slide with the color of the flower petals. Let's also talk about the fact that blood types, the ABO blood group type, is also an example of multiple alleles, because we have this IO, the O type. Now, A and B are codominant to each other, but it turns out that A is dominant to O and B is dominant to O. So if somebody is IA and IO, phenotypically, they're going to be blood type A, because A is dominant to O. Same thing with this case where you have the person who has one B allele, IB, and one O allele, IO. In this case, B is dominant over O, so they're blood type B. Just to review some terms, this is their genotype, BO. This, blood type B, that is their phenotype. Now, what I would encourage you to do, just so you get used to this, ask yourself what kind of children, what kind of blood types would there be if AO mated with somebody who was AB, or somebody who had the blood type B but was heterozygous, BO, mated with somebody who was BB. Make some predictions about what their children are going to be. I should also point out one last thing here with our ABO blood groups. And if somebody is IO, homozygous for IO, they're going to have blood type O. Sometimes there'll be a question, why don't we just do a practice here? Well, let's do it on the next slide. Sometimes I might ask you a question like, there is a child born, and the child is let's make them blood type A. Okay. So this person's genotype is either IA, homozygous for A, or IA, IO, because A is dominant over O. And I told you that his mom was also blood type A. So his mom, for simplicity right now, I'm just going to stop writing that letter I, because it'll go a little quicker. His mom is either AA or AO. We don't know which. We're only told phenotypically she is blood type A. So genotypically she could be either one of these. If the father of her baby, well, let's make this Jerry Springer-ish, okay? She has 3 people on the stage, and she says she's not sure who the father is. And let's say this first one has blood type B. The second person has blood type O. And the third person is, what should we make them? Let's make them AB. Now the question is, which one of these is the father? Which one of these is not the father? And can we be sure about any of these? Well, let's take a moment. Actually, what I'd like you to do is hit pause and ask yourself, what are the possible genotypes for this person, the possible genotypes for this person, this potential father, and the genotypes for this potential father. Okay. Go and do that and hit pause and then come back. Well, hit pause first, then come back. So the possible genotypes for this individual is either BB or B and then O. The possible genotypes for somebody who has blood type O, only one choice. They've got to be OO. Same thing with AB. Only one possibility here. It's AB. Now, think about this. What kind of gametes could this person make, and this person make, and this person make? Again, go and hit pause and then hit play when you figured it out. The possible gametes this person could make would be blood type B, and if they happen to be a genotype BO, they could also make blood type O. And over here, they're only going to make blood type O. And over here, they're going to make blood type A and B. So 2 possible gametes. Now ask yourself, can any one of these be the father? Can any one of them be excluded? And the best way to do this is just set up a whole bunch of Punnett squares and see if any of those combinations could give you blood type AO. Go ahead and hit pause and then come back. Okay. First of all, the child here, right here, is either AA or AO. And let's just say we don't know which one and we're not going to find out. Based on that, could this person be the father? The answer is yes, they could be the father, because they could have contributed an O allele to this child, this allele, and the mom would have contributed an A allele. What about this individual? Well, this male has to contribute O, and then mom would have had to contribute A. All right, what about this person here? Could they be the father? They could be the father if the child is this genotype, AA, where mom gives them an A and dad gives them an A. Or they could be AO if mom was AO and she gave the O and dad gave the A. All right, so right now all 3 of these guys are on the hook. We don't know which one it is. Let's say they tell you that it turns out that this child has a fraternal twin that is the same father. And we're going to assume it's going to be the same father. And let's say the blood type of this child is blood type B. So the second child is blood type B. So they could have a genotype of either BO or BB. We don't know. All right, now ask yourself, can any of these fathers, potential fathers, be excluded? Remember, mom, in this case here, has to contribute an O. So this case here, we have now just confirmed that mom is AO. She has to be. If she's AA, then this child couldn't be hers. And we're pretty sure it's hers because she gave birth to him. All right, so with that knowledge, mom has to be AO. Which one of these fathers could still be the father? Well, let's look over here. What we need from dad here is the B allele. So it's got to be, well, it doesn't have to be this person, but he's still in the running. What about this person here? I think they're off the hook because they have to contribute an O, and we know if this person is BO that they had to get the O from mom here. Okay. So he is off the hook. I don't know if he's happy or sad. What about AB? Could he be the father? Okay. Now we know that mom, right? We just determined that mom has to be AO, so she can't be AA. We could also make an assumption now that the baby here, the child here, has to be BO. He can't be BB. Why can't he be BB? Well, the only way you could be BB is if both mom and dad contributed a B allele. She can't give it a B allele. So we know that the baby has to be BO. All right, so that means that this parent here would have to be able to contribute a B allele. And yeah, they make A and B alleles. So this person is still on the hook. All right, now, how are we going to determine who the real father is? Well, you could do some more genetic tests. In fact, anymore these days, they don't use blood typing to determine paternity. But for the sake of this argument, let's just say, because we're on Jerry Springer's show, let's just say that there's a third child. It's fraternal triplets. So it has to have the same father for these three children. And let's say this baby is blood type O. So its genotype has to be OO. One of these guys is going to be the father. We're going to be able to figure it out now. This guy's still out of the running. If we didn't have baby 2 here, he would still potentially be the father because he's producing O gametes. But we know he can't be the father because of baby 2. All right. Now, which one of these two fathers, this one or this one, could contribute an O allele? Because remember, genotypically, this baby is OO. So one of these O's comes from mom, and we already know she's AO. One of these comes from dad. This dad is AB. There's no way he could give an O allele. This father's either BB or BO. So we've just eliminated AB. And so this guy here, hopefully he's happy because he's just discovered he has 3 children. And hopefully he's got a good paying job too. Or she has a good paying job and he can stay home and raise these kids. I don't know. Now, we could carry this on a little bit more, but we're not going to. But if you understand this, then you understand how to do Punnett Squares well. You understand how to make predictions. There's often a question on the test that's going to force you to think about it this way. So if you want to have practice, just kind of make up one of these scenarios where the baby has a certain genotype and the mom has a certain phenotype and the mom has a certain phenotype, and then ask, what does the father have to be? Okay. Well, that's it now for, we've done complete dominance, we've done incomplete dominance, we've done codominance, and we've done multiple alleles. And then we talked a little bit about blood groups here. Okay, let's move on to some other extensions to Mendel's rules. Up to now, we've talked about essentially one gene causing one mutation. Now, there might be some variability in how that mutation is or how certain genes are expressed. But now let's talk about a couple other terms here. The first term I want to talk about is called pleiotropy. And what pleiotropy means is that you have a single mutation, so single mutation in one gene, that means. It could be recessive or a dominant mutation. Typically, they're going to be recessive, though. So a single mutation in one gene. And unlike everything we saw before, remember we saw if you have a mutation, you change the color of the plant, the flower, the shape of the seed, the blood type, all those other things. But it's a single gene causing a single phenotype. Now with pleiotropy, again, single mutation, but it causes many phenotypes, different phenotypes, not even necessarily the same organ or tissue. So a classic example of pleiotropy, there's several good examples, but a nice example is cystic fibrosis, and I'm just going to abbreviate this as CF. Cystic fibrosis, most of us are familiar with the main symptom of cystic fibrosis, and that is to do with your lungs and fighting off infection. But the reason this damages the lung tissue is because of an ion balance in the membrane. And so any tissue where there's ion balance is important. When you have this gene mutated, you have the potential of causing an altered phenotype in those tissues. So for instance, it also affects the pancreas, sweat glands, very much so in the sweat glands. In fact, that leads to one of the earlier ways to diagnose it. Sometimes people notice a very salty taste on, say, maybe their child's skin when they been over to kiss their child on the forehead or something like that. Sometimes they'll come back with a very salty taste, and that's kind of a big sign. Another phenotype that is often not noticed, or I would say hadn't been noticed until maybe recent years, and that's infertility. And the reason it hadn't been noticed until fairly recently, I don't know when it was first diagnosed, but is infertility. And the reason that it took a while before this got recognized as a real phenotype is that up until recently, somebody with cystic fibrosis didn't necessarily live to the age of sexual maturity. And so as a result, you never knew that the person was infertile because they usually didn't live long enough to know that. But now that treatment has become better, we still can't cure cystic fibrosis, but people with cystic fibrosis can live longer now. And so now they do reach the age of maturity, sexual maturity. And so sometimes we are recognizing that there is infertility. And the infertility is mainly seen in males. All right, now let's think of another variation of this. All right, now let's look at another term. And this term is polygenic. Polygenic is something we mentioned in class the other day, but let's spend a little bit of time talking about it right now. It is, in many ways, you could think of it as the opposite of pleiotropy. Pleiotropy, as we just said, is one gene mutation gives you many different phenotypes. Polygenic is many genes are necessary in the same phenotype. So many genes function in the same phenotype. So you might have, well, I'll give you some examples here in a moment. This is probably more the norm than the exception. Many traits in humans and other organisms are polygenic. More than one gene functions in the same trait. We mentioned a couple in class the other day, and so let's just go and write a couple down now real quick. One was eye color, another was skin pigmentation, and many others. I want to focus on skin pigmentation mainly because we did talk about this in class the other day, but also because there's this nice little figure here I want to go over, because I think it illustrates it nicely. Okay, so let's look at this specific example of skin pigmentation. It's a really nice illustration. You can see how, if we look at these two parents here, this one here and this one here, they have about the same skin pigmentation. In fact, let's look at them and see if we can find them down here in this chart. They're right here. Okay. So they're not fully recessive like you'd see here with the lightest pigmentation, and they're not fully dominant like you might see here with this skin pigmentation. But you could see how many different ways they could make their gametes. So if this was the sperm, say from this first parent here, the male parent here, you could see how they could make little a, little b, little c gamete like you got here, and these different other combinations. Big A, little b, little c, little a, big B, little c, little a, little b, big C, and down the line. This parent here, genotypically they're the same, so they're going to make the exact same gametes. But you can see how they would combine those gametes in all these different ways here. And you can see the vast variety in skin pigmentation that they could get. They could get a child that is of much lighter skin pigmentation than either one of them. They could get a child who is at the other extreme, a much darker skin pigmentation than these two parents individually. Even in these rare cases here, right, this would be a 1 in 64 chance and 1 in 64 chance. So pretty rare, but it's even more rare because the number of genes involved in determining the pigmentation in somebody's skin is much more than 3. So it's extremely rare to get these right combinations, have the right parents with the right ability to make the gametes, and then to get these different varieties. You can see that most of the kids are going to be, say, right around here, 20 out of 64, almost 30% of them, are going to have about the same color skin pigmentation as the parents. But you do get these extremes. I don't have a good figure to show you, but the exact same thing occurs with eye color. These polygenic traits will provide many different various phenotypes in the offspring. Next, let's talk about multifactorial. And what multifactorial means is that any given trait that is a multifactorial trait requires both a genetic component and an environmental component. I just abbreviate environmental. For any given trait, sometimes it's stronger on the genetic side, and sometimes on other traits it's stronger on the environmental side. We won't have a chance to talk about it in this class, but a great way to study which one's more important, genetics or environment, is to look at the differences in fraternal twins and identical twins. You'll have to take genetics to learn a little bit more about that. Or ask me in class, perhaps. One example shown here in these two pictures here, this is a plant, this is a specific flower called a hydrangea. Now these flowers, both of these here, they're the exact same genotype. And so let's just say that they are both, we'll use big A as their genotype for their two alleles. So why are they different colors? Well, it turns out that the color of the flowers changes depending upon how much aluminum is in the soil and what the pH of the soil is. And that will have a difference upon the shades of the different colors of flower. We also have many examples of multifactorial traits. We see them in humans a lot of things such as, intelligence, athletic ability, even diseases like diabetes and heart disease are truly multifactorial traits. Well, I should say diabetes type 2. These are all very important multifactorial traits, meaning that both you need the right genetic combinations and the right environment to give you different phenotypes. All right, let's move on to this very last topic. And it probably won't take too long to talk about this one, but we need to introduce it, and then we'll spend a little bit more time talking about it tomorrow. And that is understanding pedigrees. So let's just start drawing one here. Circles are always female in pedigrees, and squares are always males. So let's just go and write over here, just to help us, just to help remind us. Squares are males, circles are females. All right, we draw a line in between these to indicate that they have children, and then we draw their children down like this. And let's just say that this particular couple had, let's give them 4 kids. We'll give them 2 boys and 2 girls. I'm drawing the girls first, not for any particular reason. And this would be the next generation. If we want to use our terms we've used before, this is the P generation, this is the F1 generation. This son here, now he may marry somebody from a different family. let's hope he marries somebody from a different family, right? Then they'll have children. And let's just say they have 2 children. We'll give them 2 daughters. That'll be fine. Now we can tell a lot about a specific trait by looking at a pedigree. So let's just look at this simple pedigree here. And let's make it a pedigree of an autosomal recessive trait. And let's make it, oh, we've already talked about cystic fibrosis, so let's go with cystic fibrosis, CF. Remember, CF is a autosomal recessive trait. So you need 2 little c's in order to have the trait, and at least 1 big C allele to be healthy. And then little c is a person that has cystic fibrosis. Okay. One of the clear signs that you're dealing with an autosomal recessive trait is if you have 2 parents who are not affected, but they have a child who is. So for instance, you might have these 2 parents here. They don't have the trait, but let's give the trait to 2 of their kids. Well, let's just give it to 1. We'll give it to this second daughter. And to show that somebody has a trait, you color it in all the way. So just by looking at this pedigree, we can see the parents are not affected, but this daughter is. Now, we don't know about this daughter or these 2 sons. We know they don't have cystic fibrosis, but they could be carriers. But one thing we do know from this pedigree is that the 2 parents must be heterozygous. And let me explain to you why we know they have to be heterozygous. Let's draw Punnett Square down here. For instance, let's see here. We know one of the daughters, in this case it doesn't matter if it's a daughter or not, is little c, little c. The only way she could be little c is if mom and dad both gave her a little c allele. It's the only way she could have it. But we also know that they're not affected. So that means they have to be heterozygous. They have to have 1 big C. And so that means here, 1 big C, 1 big C, like so. And so we know there's a 25% chance that they will be affected, have a child that has cystic fibrosis, a 50% chance of having a child that is heterozygous or a carrier, and a 25% chance that the child is homozygous dominant. Now, we can't tell just by looking at this child, or this child, if they're a carrier or if they have the disease. But one thing we can tell, or one thing we might be able to determine, is if one of these children has a child with cystic fibrosis. So for instance, if this male son here in the F1 generation had children and one of them was affected, so let's make this daughter affected, then we know, well, let me ask you, and then you can hit pause while you think about it. We know she must be little c, little c. What does that tell us about these 2 parents? So go ahead and hit pause and think about that for a moment and come back. If she is little c, this daughter, then we know mom, or I should say dad, and mom have to be carriers for the same reason. Often, when you think of autosomal recessive traits, people say it skips a generation. All that means is what we see right here. This generation's not affected, so it skipped this generation, and now this generation is affected. These 2 aren't affected, but this child is. That's all we mean. It should also be clear, because often it's misinterpreted. It doesn't mean it has to skip a generation. It just means it can skip a generation. For instance, a dominant trait, and let's pick, for a dominant trait, do I need to redraw this? Yeah, I'm going to redraw this. And let's talk about what a pedigree for a dominant trait was. I'm going to keep it on the same slide just so we can see them side by side. And so these extra letters here, I hope it's not confusing. But let's take Huntington's disease. It's a classic dominant trait, HD. And all that means is if it's dominant, it's kind of the reverse of what we see up here. If you're little h, you're healthy. But if one of your alleles, and it only takes one, is big H, then you have Huntington's disease. It's a dominant trait, so the dominant allele, and you only need one of them, will give you the disease. So let's redraw a pedigree, and we're going to use the same kind of boys and girls mix here. So let's just redraw this exact same pedigree. So here's mom, here's dad, and again they have children. Okay, now let's say what happens here is you notice that this, let's have the male affected here. So we're not always picking on the girls as we did in the last slide, or the last pedigree. So this son here, it should still be a square, has Huntington's disease. If this child has Huntington's disease, we know that one of these two parents has to have it, because we know that one of these two parents gave him that big H allele. We know that he is, and let me say this in a slightly different way. We know he must be a big H, little h. And you may ask, why do we know he's not big H, big H? Most dominant traits exist as heterozygous. Most dominant traits that are homozygous are lethal. A big H is lethal. They would never survive birth. So he's big H, little h. He must be. So that means one of these two is a carrier. We don't know which one, though. We don't know who gave it to him. We're going to finish filling in this pedigree here. Oh, actually, we don't really know. Because all we know for sure is that one of them is big H, little h. We know that, so if this is big H, little h, one of these has to. And we know which one it is because they're actually going to have it, right? So let's give it to dad. Dad's going to have it here, so he's going to be affected. We know one of them has to be affected. So dad has it. You can see here how it does not skip a generation. It can't skip a generation because it's dominant. If somebody has it, they can't carry it and not be expressed. So dad had it here, he gave it to his son. This son doesn't have it. He will never be able to produce a child that has Huntington's disease because it was not passed down to him. And we know it wasn't passed down to him because he doesn't express it. So he must be little h, little h. All right. Now, your book goes through several examples of recessive and dominant traits, autosomal and recessive traits, and autosomal dominant traits. Go ahead and look through those, and you should be comfortable with several examples of each of these as was indicated on the study guide. For right now, that's all we're going to talk about for Chapter 14. I'll see you in class. If you have any questions, let me know. Bye.